The story, as it's been told, goes like this. A Soviet research vessel in the Southern Ocean, the Akademik Fedorov, according to the account, is running routine biological survey trawls in water cold enough to slow most chemistry, when a net comes back from depth holding something the crew can't file under anything they know. It looks like a spider. It moves like a spider. But spiders don't live on the bottom of the Southern Ocean, and they don't come like this. A body so reduced it looks drawn in wire, 8 legs jointed like a skeleton's fingers, each one longer than any reasonable body should require. When they stretch those legs to measure them, flat on the deck, tip to tip, the span approaches 30 inches. 30 inches of leg on an animal whose central body you could cover with your palm. Nobody aboard had a category for it. It wasn't a spider, it turned out. What they had pulled up was something older and stranger, a creature that breathes through its knees, carries its gut in its limbs, and has been doing both of those things for longer than vertebrates have had jaws. The water it came out of was exactly the reason it could exist. Because the Southern Ocean isn't generically cold the way the deep sea is cold. It's cold in a specific way, oxygenated in a specific way, and ancient in a specific way. And those 3 conditions, stacked in the same place, build the animal on the end of the net. The giant Antarctic sea spider isn't an anomaly. It's proof of concept, an animal whose impossible proportions are made possible when cold water removes the size ceiling, and whose body is a walking physics demonstration of what happens when that ceiling lifts. The body itself is where the size begins to make sense. A sea spider's trunk isn't a body in the conventional sense. It's a narrow junction, a short, almost featureless hub from which the legs extend in all directions. The genus name, Colossendias, translates roughly as colossal legs, which captures the proportion problem in 2 words. In the largest specimens of Colossendias colossea, the species formally described by Wilson in 1881 and recognized by Guinness World Records as the largest sea spider, the leg span reaches approximately 70 centimeters, about 27 to 28 inches. Legs the length of a forearm and a half, attached to a central body the diameter of a large coin. The members of the family Colossendeidae that make up Antarctic deep-sea trawl catches are described in the scientific literature as having leg spans frequently ranging from 40 to 50 centimeters, the most conspicuous component of any Antarctic trawl haul precisely because of scale. The body is so small that the major organs simply can't fit inside it, so they went into the legs. This isn't metaphor. The midgut sends long tubular extensions, diverticular, branching down into each walking leg. In most species, these branches reach past the femur and well into the tibia. A micro-CT atlas of Nymphon gracile shows the gut diverticulum extending as a developed lumen to the distal end of the second tibia, then continuing as a compact cell strand almost to the junction of the tarsus and propodes, nearly to the leg tip. Separate gonadal tissue runs alongside. The gonopores, the openings through which eggs and sperm exit, sit on the second coxa of the walking legs, not on any central body structure, and in many species are repeated across multiple leg pairs. Anatomy textbooks are direct about this. Sea spider bodies bear 4 pairs of long, spindly walking legs, into which gonad and midgut diverticula extend. Run your eye along one of these legs, joint by joint, and you are looking at a digestive organ and a reproductive organ simultaneously. A 2024 chromosome-level genome assembly from the University of Vienna confirmed their phylogenetic position at the highest resolution yet. Sea spiders, Pycnogonida, are understood to be the sister group to all other Chelicerata. Now comes the question that makes biologists stop. How does it breathe? Sea spiders have no gills, no lungs, no tracheal system, no specialized respiratory pigments, no hemoglobin, no hemocyanin, no molecule doing the oxygen transport work that those proteins handle in nearly every other arthropod. The World Pycnogonida database states it bluntly. Sea spiders have no gills, no lungs, relying entirely on cutaneous respiration for their O2 needs. They exchange respiratory gases through a dry cuticle. The exoskeleton is the gas exchange surface. Micro-CT imaging reveals it's porous, riddled with cuticular openings that provide pathways for diffusion inward. Samuel Lane, completing his doctoral research at the University of Montana, measured oxygen flux through these pores directly. His 2018 paper in the Journal of Experimental Biology confirmed pore-mediated diffusion and showed that modeled oxygen flux through the pores closely matched measured metabolic consumption across a wide range of body sizes. The pores aren't decorative, they are the entire respiratory system. The problem that creates is internal transport. Diffusion across an outer surface gets oxygen into the outermost tissue. Getting it to the gut diverticulum running through the core of a leg requires something more. In 2017, H. Arthur Woods and colleagues published a paper in Current Biology describing what that something is. Sea spiders use peristaltic contractions of the gut itself, the mechanical wave motion normally associated with digesting food, to circulate oxygenated fluid through the leg branches and back through the body. The gut functions simultaneously as a digestive organ and a circulatory pump. Experimental work using oxygen electrodes placed in leg fluids confirmed that gut contractions move oxygenated fluid around the body and that contraction rates increase under hypoxic conditions. The University of Hawaii press release for that work chose the clearest possible summary. Sea spiders use their legs as gills and their guts as hearts. That isn't a simplification, it's the mechanism. One more detail about the cuticle. Shishido's 2019 research at the University of Hawaii at Manoa showed that cuticle porosity in Colossendice megalonyx increases as animals grow larger. A bigger spider has a more porous exoskeleton, not by design, but because selection has favored animals that could maintain oxygen supply while growing. The body expands its capacity in parallel with its size. This is part of why the oxygen ceiling doesn't hit immediately. Feeding is through a muscular proboscis, a triradiate tube at the animal's front end, ringed with 3 lips and an internal filter that shreds prey material before it enters the midgut. The animal inserts this proboscis into prey tissue directly: hydroids, bryozoans, soft cnidarian flesh, sponge material. Wide proboscised species go after sea anemones. Extremely narrow proboscised species thread through the frontal wall pores of bryozoan zooids. Reproduction adds one more anomaly. In all sea spider species without exception, the males carry the fertilized egg masses externally, attached to specialized appendages called oviges, extra limbs positioned between the walking legs, coated with cement glands in the femora that bind the eggs in place. Males have been documented carrying up to 14 egg masses simultaneously. In temperate waters, off Washington State's Friday Harbor, for example, sea spiders have leg spans of around 1 centimeter. Antarctic species of Colossendice routinely reach 30 centimeters or more. The largest individual in H. Arthur Woods's 2008 McMurdo Sound study was Colossendice australis, weighing 19.5 grams with a leg diameter of 4.2 millimeters. The smallest in the same study was Pentanymphon antarcticum at 0.02 grams. 3 orders of magnitude in body mass, same family of animals, same seafloor. The comment that every sea spider video receives is predictable. That's not actually a spider. It deserves to arrive in the narration before it arrives in the comments. Sea spiders aren't spiders. They aren't arachnids. Spiders belong to Arachnida, which sits inside the larger branch, Euchelicerata. Sea spiders, pycnogonids, split from the chelicerate family tree before Euchelicerata evolved. They are the sister group to all other chelicerates, the oldest branch, not subsumed into the groups that developed later. Multiple phylogenomic analyses, including a 2021 dataset published in Molecular Biology and Evolution, drawing on dozens to hundreds of genetic loci, consistently place pycnogonids in this basal position. A 2025 chromosome-level genome assembly from the University of Vienna confirmed it at the highest resolution yet. Sea spiders, Pycnogonida, are understood to be the sister group to all other Chelicerata. Scorpions, mites, horseshoe crabs, ticks, and true spiders are all, in evolutionary terms, newer architecture. Sea spiders are what preceded them. The fossil record goes back to Haliestes dassos, found in volcanic ash deposits of the Silurian age Herefordshire Conservat Lagerstätte in the UK, approximately 425 million years ago. Civita and colleagues described it in Nature in 2004. The specimen was reconstructed slice by slice, ground away microns at a time, digitally imaged, and assembled by computer graphics. The animal looked, functionally, like a modern sea spider. Same proboscis, same seafloor lifestyle, same basic body plan. Remarkable morphological and ecological stability is how the paleontological literature describes this lineage across geological time. About 1,350 described species are alive today, distributed across every ocean floor from the intertidal to abyssal depths, tropics to poles. In tropical and temperate waters, adults have leg spans measured in millimeters to centimeters. Only the Southern Ocean grows them to 70-centimeter spans and record dimensions. Same ancient lineage, radically different scale. That contrast is the engine of everything that follows. In 1999, Gautier Chappell and Lloyd Peck of the British Antarctic Survey published Polar Gigantism Dictated by Oxygen Availability in Nature. Their method, body length data for 1,853 species of benthic amphipod crustaceans from 12 global sites, correlated with potential oxygen availability at each site, calculated from water temperature, which governs dissolved gas solubility. The maximum 5% of body sizes at each locality tracked oxygen availability closely. Their conclusion, maximum potential body size is limited by oxygen availability. The paper has accumulated more than 378 citations. The physical basis is established chemistry. Cold water holds more dissolved gas than warm water, governed by Henry's law. At 0 degrees Celsius, water at saturation holds roughly 14.6 milligrams of dissolved oxygen per liter. At 10 degrees Celsius, that falls to 11.3. At 25 degrees Celsius, 8.3. Polar surface water holds between 40 and 70% more dissolved oxygen than tropical water at comparable saturation. For an animal breathing through its skin, ambient oxygen concentration is the immediate ceiling on body size. More oxygen available per unit of cuticle surface means larger bodies can be adequately supplied. Cold does a second thing simultaneously. It suppresses metabolic rate. An Antarctic sea spider at negative 1.5 degrees Celsius has substantially lower oxygen demand per gram of body mass than the same animal would have at 15 degrees Celsius. Supply goes up, demand goes down. The ratio between them, the effective budget for body size, shifts dramatically in the animal's favor in polar water. This is the oxygen temperature hypothesis. Cold increases oxygen availability and decreases oxygen demand, releasing the size constraint that diffusion-limited animals face in warmer seas. It's the leading explanatory framework for polar gigantism, and it's built on sound physical principles. But it isn't settled science, and for sea spiders specifically, it has been directly tested and found complicated. In 2007 and 2008, H. Arthur Woods and Amy Moran led a study at McMurdo Sound using 49 pycnogonids from 12 species, spanning those 3 orders of magnitude in body mass, from 0.02 grams to 19.5 grams. Animals were tested at 3 oxygen concentrations: severe hypoxia at 17.6% of air saturation, moderate hypoxia at 43%, and normoxia at 92%. The oxygen hypothesis predicts that large animals, operating closer to their size ceiling, should fail disproportionately under low oxygen conditions. The data showed no such interaction. Large pycnogonids and small pycnogonids degraded in performance equally as oxygen declined. These data don't support the oxygen hypothesis of Antarctic pycnogonid gigantism, the paper concluded, and suggest that explanations must be sought in other ecological or evolutionary processes. A 2020 synthesis by Moran and Woods in Integrative and Comparative Biology identified at least 8 proposed mechanisms for gigantism and concluded the picture remains genuinely unresolved. Temperature is a strong causal agent. The specific mechanism in sea spiders hasn't been isolated. What this means practically, the oxygen temperature framework is the right context for understanding why the Southern Ocean produces giants. The physics are real and documented for amphipods and multiple other invertebrate groups. For pycnogonids, the relationship is more nuanced, partly because these animals have already engineered around the diffusion problem in ways other animals haven't. Gut peristalsis, scaling cuticle porosity. Oxygen availability still matters, but it may enable giant size in sea spiders rather than directly limit it in the way the original hypothesis predicted. The honest position is the one the literature takes. This is a leading hypothesis, not a concluded mechanism. Now for the question most discussions of polar gigantism skip past. What is actually different about the Antarctic compared to the deep sea? The deep sea is cold. Animals get large down there too. Giant isopods in the genus Bathynomus can exceed 40 centimeters, giant squid, giant amphipods. So why does the Antarctic specifically produce record holders? And why does the sea spider, living on the continental shelf at depths of tens to a few hundred meters, beat the abyssal giants? The answer is oxygen availability, but the geography of that oxygen is what matters, and the geography is completely different between the two environments. Polar gigantism and deep-sea gigantism are formally recognized as distinct patterns. Both involve cold. Cold isn't the whole story. The mid-ocean water column isn't uniformly oxygenated. The oxygen minimum zone, the OMZ, sits between roughly 100 and 1,500 meters depth across much of the world's ocean. NOAA's Ocean Explorer defines it as areas of low oxygen saturation persistently between 100 and 1,500 meters depth. In OMZ cores, oxygen concentrations can fall below 2 milligrams per liter. Some tropical Pacific OMZs approach near anoxic conditions. Biological consumption of oxygen by bacteria decomposing organic matter sinking from above outpaces resupply. For an animal that depends on ambient dissolved oxygen diffusing through its cuticle, the mid-ocean water column isn't a place to grow large. The oxygen budget actively works against size. Deep-sea giants living below the OMZ in the abyssal zone exist in somewhat better oxygenated water, but that water may have been isolated from surface oxygen replenishment for decades or centuries.
The Southern Ocean operates differently at the level of its circulation. Antarctic bottom water, AABW, forms on the Antarctic continental shelf through a specific process. Surface water, already cold and nearly oxygen-saturated from contact with the atmosphere, becomes denser when nearby sea ice forms and brine is rejected into the water column, increasing salinity. Dense, cold, oxygen-saturated water sinks. It flows northward along the ocean floor and spreads through the world's abyssal basins. The Australian Antarctic Division describes AABW as dense, cold, oxygen-rich water whose flow drives ocean circulation globally and assists in carbon capture. WHOI confirms Antarctic bottom waters supply oxygen, carbon, and nutrient-rich waters to the world's oceans. The critical feature of AABW is that it forms from recently surfaced water, near oxygen saturation at the moment of sinking. It hasn't been isolated from replenishment. It begins its journey fully charged. On the Antarctic shelf itself, where sea spiders live, the water is perpetually refreshed by AABW dynamics, surface contact, and the Antarctic circumpolar current, the largest ocean current on Earth, running unobstructed around the continent, driving continuous mixing. No equivalent feature exists in the Arctic Ocean, which is enclosed by continents and has fundamentally different water mass formation. The asymmetry shows up in body size data. De Breuer's analysis of amphipod size across latitudes found that 31% of Southern Ocean species had body lengths more than twice the mean size of their genus. In the Arctic, that figure was 28%. In the deep sea, 21%. In the tropics, 0.8%. Then there is time. Antarctic marine fauna has been isolated in these conditions for tens of millions of years, far longer than Arctic fauna, which was colonized from lower latitudes more recently. Millions of years of selection pressure in stable, cold, oxygen-rich, circulated water produces animals adapted to exploit exactly those conditions. The sea spider lives at the intersection of two sets of rules. The deep sea has cold, but its oxygen is either blocked mid-column by the OMZ or slowly depleted in abyssal isolation. The Antarctic has cold and reliably high oxygen on its shelves and slopes, continuously refreshed by the most vigorous surface-to-depth circulation in the ocean, maintained across geological time. Put both sets of conditions in the same place, let evolution run for tens of millions of years, and you get the animal that came up in the net. The Southern Ocean isn't just another cold ocean. It's the one place where both rules converge completely. The sea spider isn't alone in what the Southern Ocean produces. The pattern appears again across the ecosystem in distantly related lineages with entirely different body plans. In cold northern waters, the lion's mane jellyfish holds the most extreme single measurement for any jellyfish. The largest documented specimen, measured off Massachusetts in 1865, a 19th century naturalist record rather than a modern controlled specimen, had a bell diameter of 210 centimeters, 7 feet, with tentacles reaching 36.6 meters or 120 feet. A blue whale reaches 25 to 30 meters. The Monterey Bay Aquarium states the comparison directly. The tentacles of that jellyfish exceeded a 90-foot blue whale. The 1865 figure warrants the caution any 19th century naturalist record warrants. But cold water lion's mane jellyfish reaching bell diameters of 1 to 3 meters is documented in modern records, not just historical ones. These animals aren't a catalogue, they are confirmation of a policy. The Southern Ocean and adjacent cold polar waters aren't producing one oversized creature by accident. They are applying the same conditions across distantly related lineages with entirely different body plans and producing the same result.
The spider is the most anatomically striking example. Because of the gut in the legs, the breathing through the knees, the body small enough to cover with a palm, and legs that reach across a cafeteria tray. But it sits inside a pattern that stretches across the ecosystem. Why doesn't the whole Antarctic seafloor grow enormous? If oxygen supply is high and metabolic demand is suppressed by cold, what limits the growth? The exoskeleton is the answer. Shishido et al.'s 2019 paper in Proceedings of the Royal Society B, Polar Gigantism and the Oxygen Temperature Hypothesis, a test of upper thermal limits to body size in Antarctic pycnogonids, identified the constraint precisely. As body size increases, cuticular porosity must increase to maintain adequate oxygen flux. A larger animal needs more oxygen delivered per unit time through the same basic diffusion mechanism, so the cuticle must become more porous. But the cuticle is also load-bearing. It's the skeleton, the structural support for locomotion, the weight-bearing architecture of the body. There is a threshold beyond which the exoskeleton can't become more porous without losing mechanical integrity. The NSF-funded research project led by Woods and Tobalski captured it directly. Cuticle structure represents an evolutionary compromise between the competing functions of oxygen uptake and effective locomotion. The ceiling isn't oxygen availability by itself. It's the structural physics of a surface that must simultaneously let in air and hold the animal together. Temperature is how that ceiling is enforced in the present ocean. Shishido's 2019 research and the broader McMurdo Sound project found that Antarctic sea spider metabolic rates rise sharply with even small temperature increases above the normal range of -1.5 degrees Celsius.
At 9 degrees Celsius, most tested specimens were immobilized. The thermal window these animals occupy is narrow, functionally restricted to a range that the Southern Ocean, right now, reliably provides. Mathematical models built from the experimental data predict that higher water temperatures cause internal oxygen levels to decline sharply, and that the largest-bodied individuals face the steepest drop. The direct experimental tests found that large and small animals were equally affected by warming in the short term, an outcome the cuticle porosity scaling helps explain. But the models and the physics point in the same direction. Warm the water enough, and the operating conditions that built the giant body no longer hold. That warming is documented and ongoing. Curtis Deutsch of the University of Washington, in a 2015 paper in Science, modeled the combined effects of ocean warming and oxygen loss on metabolic capacity globally, projecting approximately a 20% decline in the metabolic index across much of the upper ocean. For the Southern Ocean specifically, where the conditions enabling maximum body size are cold temperatures, high oxygen availability, and vigorous AABW-supplied ventilation, those are precisely the parameters most directly affected by surface warming and polar ice dynamics. This is projection. The sea spider hasn't yet been measured shrinking. The conditions for its existence haven't yet crossed a measurable threshold. But what the oxygen physics of the Southern Ocean built, a change in those physics can unmake. The fine print that Antarctica publishes is written in conditions, not in stone. Somewhere tonight, on the floor of the coldest ocean on Earth, an animal with a body the size of your thumbprint is walking on 30 inches of leg, breathing through its knees, carrying its guts in its limbs, pumping oxygenated fluid through its digestive tract, alive because the water around it holds more of the one thing its body runs on than almost anywhere else on Earth. The crew that first hauled one up on deck, the story says they thought they'd pulled up a mistake. Something that shouldn't be there, shouldn't be that size, shouldn't look the way it looked. And in a sense, they were right to be confused. The animal doesn't make sense against most backdrops. Against the specific backdrop of the Southern Ocean, cold and oxygen-saturated and vigorously circulated and ancient, where the cold of the deep sea and the oxygen richness of a polar surface-connected water mass coincide in one place, it makes complete sense. It's, in fact, the expected result. They hadn't pulled up a mistake. They had pulled up a rule, wearing the shape of an exception. Antarctica doesn't break biology's laws. It publishes the fine print. And the sea spider, the creature that shouldn't exist, is written there in full. If you want more stories that go this deep, subscribe.